We've previously discussed how protein synthesis and degradation can regulate an overall enzymatic process. This process is relatively slow and can take hours to days. Therefore, we need mechanisms to rapidly regulate enzyme catalysis. In this short session, we'll talk about allosteric regulation and covalent modifications that can rapidly regulate, either increase or decrease enzyme activity in the short term. So we'll start first by discussing allosteric regulation. And it's important to note that allosteric regulation can influence either CHEM or Vmax. We won't focus on those specific regulatory mechanisms here. And for our purposes, we'll use the term allosteric regulation to regulate most of the enzymes we'll talk about. If we're talking specifically about a change in CHEM or Vmax, that will relate to a competitive or non-competitive inhibitor, as previously discussed. So the first encounter with allosteric regulation we've had is with hemoglobin. You can see here that hemoglobin activity can be influenced by 2,3-BPG, and in this case, it stabilizes the tense state of the enzyme. So allosteric regulation can be both in the form of activation or inhibition of the enzyme. In the case of allosteric activators, they increase the activity of the enzyme, whereas inhibitors will decrease the activity of the enzyme. An example of an allosteric activator is noted here with the regulation of our glycolytic enzyme pyruvate kinase. Pyruvate kinase is regulated in a feed-forward manner by the upstream substrate fructose 1,6-bisphosphate. The accumulation of fructose 1,6-bisphosphate in the glycolytic pathway will feed forward and allosterically activate pyruvate kinase. An example of an allosteric inhibitor is here in the regulation of fructose 1,6-bisphosphatase, and this is an enzyme we'll discuss in gluconeogenesis. It's regulated through allosteric inhibition by AMP. It's important to note that enzyme regulation by AMP or ATP is very common as it links the synthesis or oxidation of compounds with the relative energy state in the cell. In this case, the low energy state, signified by AMP, will inhibit the synthetic reaction and reduce the overall production of glucose. Enzymes can also be rapidly modified through covalent modifications, and the most common covalent modifications that we'll discuss are phosphorylation, acetylation, and methylation. Protein phosphorylation is a highly versatile selective process, and not all proteins are subject to phosphorylation. While the most common enzyme function effect is the protein's catalytic efficiency, phosphorylation can also alter the protein's location within the cell, its susceptibility to proteolytic degradation, or responsiveness to regulation by allosteric ligands. It's important to remember that phosphorylation can increase or decrease an enzyme activity, and we'll discuss those in context throughout the course. Histone acetylation is another common covalent modification. This will regulate the transcriptional activity of a specific gene locus. And finally, methylation can also increase or decrease enzyme activity. Our common methyl donor is S-adenosyl methionine, or SAM, and we'll discuss the production of this compound when we discuss methionine metabolism later in the course. So if we look specifically at protein phosphorylation and dephosphorylation, it's highly versatile. The amino acids serine, threonine, and tyrosine are primary sites of phosphorylation due to their functional groups, which have an exposed hydroxyl. The phosphorylation is catalyzed by a kinase, and this requires ATP with the transfer of the phosphate group onto the enzyme. The phosphatase, remember, is a hydrolase reaction and will dephosphorylate the enzyme. Keep in mind the major role of phosphorylation for this course will be the regulation of enzyme activity. It can also influence the location of an enzyme within the cell, the susceptibility of the enzyme to proteolytic degradation, and the regulation by allosteric ligands. Next, if we look more specifically at histone acetylation, this is a schematic showing DNA, illustrated here in this light blue color, wrapped around a histone octamer. This is how DNA is supercoiled within the nucleus. DNA is generally negatively charged, and these histone proteins are positively charged, and the attraction between those two forces allows for very tight binding. When we acetylate the histone octamer core, this will decrease the positive charge on the histones and thereby decrease the affinity of the DNA for these proteins. This allows for the unraveling of DNA from the protein association, making the DNA locus more transcriptionally active. So let's summarize again with a pathway problem type question by asking the question, if we have a protein, in this case protein Z, that requires phosphorylation of an exposed tyrosine residue for activity, what happens if we modify this tyrosine to a glycine? And what impact does that have on the activity of protein Z? Well, first of all, the loss of a phosphorylation site will reduce the activity of the enzyme. How do we know that phosphorylation site has been lost? Well, if we remember back to the structure of tyrosine, we know it has an exposed hydroxyl group, whereas glycine does not. Therefore, this modification to glycine alleviates this phosphorylation site. So it's likely that this will reduce the activity of the enzyme. Let's look at this from a specific paper to relate this to enzyme activity. So this piece of data is taken from a Journal of Biochemistry article that was looking at specific phosphorylation sites on the enzyme hormone-sensitive lipase. Hormone-sensitive lipase is an enzyme involved in lipolysis, and we'll discuss that later in the course. What you're seeing here on the y-axis is the activity of the enzyme. You can see here that the wild-type enzyme has 100% activity. What's illustrated here are modifications to different serines, denoted here as S, to an alanine. The change of the serine to an alanine alleviates a phosphorylation site, and the authors of this paper are asking the question, how does the removal of this phosphorylation site impact the activity of the enzyme? What you can see here is removal of the phosphorylation site at amino acid 563 generally does not have much impact on the enzyme activity, as you can see enzyme activity at about 100%. If we look further down here, you can see that the modification of two phosphorylation sites in combination, so the modification of the serine at amino acid 659 to an alanine, and the modification of a serine at amino acid 660 to an alanine, nearly reduces the enzyme activity to 0. What you should be able to take away from this graph is that some of these serines across the amino acid structure of this protein can be modified without consequence. However, removing of a regulatory phosphorylation site, such as illustrated here, will greatly negatively impact the activity of the enzyme. Again, for full reference, the paper can be seen here with the remaining pieces of data.